Okay, the last section of this is the detailed section of immunity, but it makes perfect sense as to why somebody would want to get vaccinated. When the body comes in contact with what are called foreign antigens, an antigen is a protein or could be considered a microbe that the body doesn't recognize. Right after a person is born, that person's immune system is not quite developed, and it goes around and inventories all the cells to make sure that it can recognize cells as being self. So it doesn't attack. So the immune system will not attack its own cells. The ones that are foreign, and that's anything, the immune system will attack that. So we're going to talk about what happens when a foreign antigen gets inside the body. All right, there are two things here. There's something called a primary immune response. What that is, that's the very first exposure to a foreign antigen. Could be anything. Could be a vaccine. It could be the real illness. I got chickenpox as a kid, and I had a primary immune response to varicella, which is the chickenpox virus. The first exposure has a longer lag time. Now, lag time in immunity means the time for the body to develop an immune response. In other words, the body's developing an army to attack this foreign antigen. You get less antibody production, and it takes a lot longer. Now, one of the things you get from these exposures, though, you get something called a memory cell. And those are for the second exposure. We'll talk about that in a minute. The secondary immune response is a second exposure to the same antigen. And I don't want to date this video, but right now the coronavirus pandemic's going on. People who are getting it and they're getting over it, they're now saying that just because somebody's had it, it doesn't guarantee that person can't get it again. So it's saying the coronavirus, this wouldn't necessarily, one of those type of microbes that this would not necessarily apply to. The secondary immune response is a much larger response, and the lag time is much shorter because a person has what are called memory cells, and they get stored in the lymphatic system. So this is a great diagram. Here is, on this particular axis is time. It says days after exposure to antigen, and then this says plasma antibody concentration. You could just look at that as plasma T cells and B cells on this particular axis over here. Here's the primary immune response. Look, it takes almost 10 days for this thing to even rise up to this level. In that 10-day period, if you had an organism that was lethal, the person's not going to make it. You can see it peaks at about, I don't know, was that day 23 here? And then it starts dropping off as far as the body's response, developing armies, and trying to get rid of an infection. The secondary response, look, almost immediate. And you see how much more in numbers going across here. Look how many more cells, white cells, armies are developed as a result of having already been exposed to something. This is the entire basis for a vaccine. Vaccines are usually the primary, well, vaccines are used as a primary response, but the vaccine has either the dead pathogen or it's called attenuated, meaning it's weak, so a person doesn't get a full-blown infection. But in the process of doing that, they get exposed to that particular antigen, and then when they have a secondary response, now you have this big giant response, and the person doesn't usually know they're even sick. Okay, and then the last part of this, T cells and B cells. This is called specific immunity. All the stuff I've covered with skin and mucus and wax, that is non-specific immunity. This is what you get as a result of these exposures. A B cell, it's a white blood cell in blood, and this is the result of an interaction. So what happens is, I don't see it there. Okay. Well, that's okay. What happens is B cells are lymphocytes. It's a white cell that's found in blood. And B cells are floating along in blood, and let's say there's a pathogen in blood that it comes in contact with. Well, that would be when it gets exposed to a foreign antigen. Okay, like I said, you have what's called a lag time, which is this. And what happens during that lag time is B cells become this thing called a plasma cell. And plasma cells are antibody factories, these little Y-shaped structures here. And antibodies go and they attach to the foreign antigens that they were produced from as a result of that first interaction. So it's very specific. And B cells are 20% of specific immunity. So yes, when you get vaccinated, you're going to get some memory B cells that are going to be stored in the body. And if you ever get exposed to that particular antigen that you got vaccinated for, they're going to react immediately. This thing here is a T cell. T cells are 80% of specific immunity. Same thing. A T cell lymphocyte is floating along in blood. When they come in contact with a foreign antigen, they become specialized. Your body produces more of those. And those T cells go back and attack that very specific foreign antigen. And what they do is they go up, there's a certain category. They go up attached to it here, and they just tear a giant hole in the cell. And then this actually has some antibodies attached to it too. This is actually a virus-infected cell. T cells make up 80% of specific immunity. So those two together protect the body from infection from very specific microbes. HIV is such a bad virus because HIV takes out T cells, and therefore lowering a person's immunity, so now they are more susceptible to other things. Okay, so the whole idea of an immunization, in just a couple of terms real quick, and we'll get to the last of this. So a person acquires the disease naturally. That's actually called naturally acquired active immunity. If you get something naturally. I got chickenpox as a kid. If you get something through a vaccination or other means, it's called artificially acquired active immunity. Still does the same thing. You just acquired that particular immunity in a different way. Artificial meaning either from a vaccine or other methods, which is usually vaccination. So, okay, last part of this section, what are called the ABO blood groups. Why is this in here? Because of antibodies. There are 4 categories of blood cells. There's type A, there's type B, there's type AB, and there's type O. This is all determined by genetics, what are called alleles. Everybody gets 23 chromosomes from their mom and 23 from their dad. And so for blood type, you get 2 alleles, one from mom, one from dad. A and B, and blood types are determined by what are called antigens. Here's a type A antigen, you see it there. Here's a type B antigen, and those are carried on genes. There's actually an A allele and a B allele. A and B are always dominant over the O gene. So the only way, this is type O blood, the only way to be type O is if you get 2 recessive traits, 2 O genes. AB, which is the rarest blood type, if you get an A gene and a B gene, then you end up with type AB blood, and AB are codominant. But for instance, if in your genetic lottery you got an A antigen gene and the O gene, then you're going to be type A blood. A and B are always dominant over O. Now, here's the other thing. And I see it here with, and I actually gave you a separate handout for this. But the way you look at this, if you're talking about doing blood transfusion, you're trying to avoid what's called agglutination, clumping of red blood cells. Here are the 4 groups. They are determined by the antigens that are present, and then the antibodies come into play. Now, on our other diagram, this one, this is what happens. I was talking about the immune system not quite being developed when a person's born. Right after a person's born, the immune system goes around and inventories all cells. If a person has an A antigen, and this is spontaneous, in their plasma, their blood, they develop what are called anti-B antibodies. If you have a B antigen, you develop what are called anti-A antibodies. They show up spontaneously. It has to do with the tissue type. If you're type AB, because you have both antigens, it kind of works like this. If the immune system sees an A antigen, you develop the opposite antibody type. If you're type AB, you end up with both antigens. So as a result of that, a person with type AB blood, they do not develop any antibodies. AB is called universal recipients. If you're type O, you're lacking these two genes that gave you the A or the B. You can see their surface is completely clear. That's a person who's type O. It's a recessive trait. You have to get two O genes. And oddly enough, it's the most common blood type. A person who's type O, in response to having neither the A or the B, they develop both antibody types. O is called a universal donor, to a point, because there's no antigens. So they can technically give blood to anyone. So here's the golden rule. You see these little, so here is an A antigen. You see how it's constructed. If I can get this a little bigger, you see how it looks like a little divot on the top. Now look at the A antibodies. See how they are naturally designed to fit perfectly into those antigens. And that's what you want to avoid when doing blood transfusion. You cannot mix the same antigen and the same antibody. You can see the B antigens. Look at the B antibodies. How their design is, it would fit perfectly right there. So that's what you're trying to avoid when doing blood transfusions. And then the Rh factor is a completely separate thing. Some people, I do not. I am Rh negative. If you are A positive, B positive, that means on the red blood cell surface, along with these antigens, and you can even be O positive, you have what is called a D antigen, and it's called the Rh factor. It's in honor of the rhesus monkey, which is used for studying and experimentation. That particular primate and humans have the same antigen, some, on their red blood cell surface. 87% of the population has the D antigen, which would be 13% does not, which is what I'm in. So there's a whole separate video for lab for that, and I'll go over all that with you. So last thing, we've already covered cancer, just a couple of terms. It does mean unregulated multiplication of cells. There are two different kinds. We did cover this: benign tumors, malignant tumors. Classifications: carcinomas are epithelial cells. They divide very quickly. Sarcomas, leukemia is a cancer of white blood cells. Lymphoma is a cancer of the lymphatic system. Sarcomas are connective tissue. And then these are the stages. Stage 1 cancer is when you want to catch it. It's barely starting. Stage 2 may have metastasized. Stage 3, the ability to spread. Stage 4, a lot of times, is irreversible. And that means it's spread all over the body. But there are certain cancers now that can be reversed from stage 4. And those are thought to be some of the causes of cancer: ionizing radiation, microbes, or a carcinogen of some kind. So we've kind of already covered this before, but that's just a little bit about, the last little part on cancer. All right, thanks.